So next up, we have Steve and Jerome. And hi Steve. I've had the chance to chat with them a couple of times over the last year. Those were very interesting conversations. So Steve Jerome is Director of Hit Discovery at Schrodinger. During his PhD at Columbia University, he contributed to tools for molecular docking and protein structure refinement. After Columbia, he joined Schrodinger as a scientific developer working on the GLIDE team. In his current role at Schrodinger, he directs the development of a broad portfolio of state-of-the-art computational tools for small molecule hit identification. The title of Steven's talk is Active Learning for Absolute Binding-Free Energy Calculations in Drug Discovery. And with that, I hand it over to you, Steve.
Thank you. All right. Well, thank you so much for the invitation to speak. Really enjoyed the talk so far. I've already asked a whole bunch of questions of the speakers. Find them to be very interesting. And also, I'm glad that I chose this topic. We're also working in a number of areas that were talked about today, such as the remarkable space of DEL libraries and what we can do, particularly with sophisticated machine learning models, but also the integration potentially with traditional structure-based tools that are sort of underpin a lot of traditional molecular modeling. And of course, we remain very interested in the future of structure-based prediction, both of protein structures and also protein ligand complexes. So very, very much enjoyed the talk this morning. And we're actively testing a lot of those approaches. But today I wanted to talk about something quite a bit different. So today I think we've talked a lot about the limitations, the excitement, the potential of machine learning. But something that we at Schrodinger certainly believe is that it's certainly, we don't necessarily have to go down the road of AI as our sort of complete computational approach to drug discovery. We have other tools, and to what extent could those tools complement each other? So today specifically, I'll be talking about absolute binding free energy calculations. They represent what I believe, working in this field for many years, is very likely the first reference method for hit discovery, first reference-based, physics-based method for hit discovery. And I'll show you some of the data that gets us very excited about that, specifically how we're able to order and test fewer compounds to get to hits. But that's really just the beginning of where I think this technology could take us if we're able to deploy it at massive scale. All right. So we've been doing virtual screens for a very, very long time. The kind of virtual screen I'm talking about, massive library, right? We have, whether we're using a ligand-based approach, right? So that might be traditional QSAR. That might be looking at, shape screening, whether it's something like Schrodinger or rocks. Machine learning. Specifically, I'm talking about here 2D models. And it's something that's frustrated me for a very long time, that 2D still seems to be the best tool that we have, right? That we have yet to really make a significant impact with 3D-based methods, even though we think to ourselves, there's so much information here in all of this rich structural biology. Surely there has to be some way to leverage this, to predict and drive potency in our target. So what I have here is a table of just recent campaigns sampled from a decade, maybe even 2 decades, really, of drug discovery projects. And, they're modest numbers, right? So, sometimes you see hit rates that are much, much higher than that reported from virtual screening. This is what I would call a fraction of useful hits, right? So, our teams are typically working on very competitive targets where there's a competitive landscape, pursuing a sort of best-in-class strategy. So there's a lot of things out there already. So what you're trying to do, at least from the very beginning, is identify some hits that can be, that really pass, certain quality filters, but also are novel, also give you the handles to develop into lead compounds. And so by the time you do that, I've had screens where, we spit out all of these hits, And I say, well, what do you mean the hit rate was only 1%? And they said, well, we couldn't use just about anything you gave us. Right? So this is meant to really quantify what I would say is more of a true hit rate. Useful hits coming out of a virtual screen. And the average of 1%, right, what does that say? It means we order 100 compounds based on recommendations from all of these techniques, ML included, and we're wrong 99% of the time. Right? So that's a lot of resources going to waste. We know the cost to acquire, synthesize, purchase, and test these compounds, not to mention the lead time, is very significant, right? So when you're up against deadlines, you have to ask yourself, well, looking at a table like this, I really hope if I get 4 hits that one of these is useful to me, right? And we know so much really depends on getting a good hit out of the gate. So why doesn't this work better? Lots and lots of different reasons. But one of them, I think, is that our high throughput methods really aren't very accurate. They really aren't telling us enough. So one of them, of course, is traditional docking, which I'm sure lots of people are familiar with docking. But I'm talking about the rigid receptor variant. We have a single, basically frozen snapshot of the receptor, and we're basically going to predict the binding conformation of the ligand. And then we'll do an analysis of the interactions and the energetics. We'll use that to predict whether or not something's going to be a hit. Very noisy. We deal with the fact that we only have this rigid protein structure. Of course, proteins move, right? So how realistic is that really? Lots of errors. We have to make compromises on accuracy, how rigorous our screening, the quality of our screening is, because we need something to be fast and cheap, because we've got 1 million, 10 million, potentially 1 billion compounds to evaluate. And then what about ligand-based methods? Well, to extent, we know that if you have some hits and you want to find more hits that look like those hits, there are lots of ways of doing that. But when the task is to come up with something new and novel, we have found that these don't really tend to offer us the kind of value that we're looking for. And so most of our hits actually are coming from docking. And so, yes, we can make marginal improvements to this method, but is that really ever going to get us to, a big leap in performance and expectations? And we've come to the conclusion that it's basically never going to happen. So we have had change, though. Since 2021, we've had a new tool in our arsenal. And that tool is absolute binding free energy calculations, right? So you may know about FEP. Of course, Schrodinger has played a big part in the development of scalable FEP technologies. If you go back to 2015, we published a landmark study really on accuracy on a variety of congeneric series with our relative binding for energy method. We're talking about changes to a particular molecular scaffold, modest changes, right? FEP, the method, goes back decades. Not too long ago, it was a PhD project just to be able to do a very modest change. Now we delete entire scaffolds, grow new scaffolds, do all sorts of things. And the latest addition, and one that really makes obvious sense when you consider this problem, is to go from nothing, right, to the molecule and predict that. So no longer are we talking about a relative change in binding affinity. We're talking about predicting delta G of binding for a particular compound. And of course, when you have hits from screening hits that are very diverse, you might not have references, right? So this really, for the first time, gives us a way to apply FEP to the outcome of our virtual screens. So our method, the preprint for this is up already. I'm sorry, it should be referenced. I'll try and add that to the slides if they're going to be distributed. Our method here is based in a large part on existing technology. We're building on the work of Borsch and others in order to be able to do these simulations. And now, of course, with the access to massive GPUs, we're able to actually run very large numbers of these simulations. I'm not going to spend a lot of time going into detail of our method here, but the basic idea is we're going to basically annihilate the ligand completely and individually. That means for the purpose of the simulation, it has no interactions with anything, not even itself. We're going to use cross-linked restraints to basically position the ligand exactly where we believe it should be. And of course, that step is very dependent on successful prediction of the initial pose. Right? So there's some local sampling, but if you predict the pose completely wrong, this method will produce an incorrect result. And because the free energy of these restraints, the restraints are chosen so we can compute them analytically, we know exactly what that term is, and we can easily account for it in this thermodynamic cycle. So how does the accuracy of this compare to relative FEP? And this, I have to say, kind of blew me away. So this is the same data set I mentioned from 2015, very modest by today's FEP technology. Right? This is kind of like, well, if you can do this, you ought to be able to do this. It's kind of a baseline test, right, of sort of basic perturbations that you might do in drug discovery. Now, imagine for RBFEP here, we have, for every one of those compounds, this is a series, and we're using a very related scaffold, and we're just computing the change, right? So it's a very small perturbation. But here we are in absolute binding FEP, deleting the entire ligand, right? So the whole ligand, essentially we're asking for this alchemical free energy method to predict that delta G starting from essentially nothing. And we're able to get very close to the performance on this data set. Now, it is more computationally expensive, which is where active learning really plays a role. OK, so let's see how this affects virtual screening. So there was this nice paper on JAK2 published by Jorgensen and coworkers where they were assessing different rescoring methods. So you can imagine that's kind of where I'm going here, right? The assumption being that, OK, well, the raw docking is kind of crummy, but maybe we believe it's giving us some enrichment, right? Some enrichment. Now, so we run this big screen. I mean, big, not necessarily by modern standards. 4 million compounds are using GLIDE. Now we're going to look at the number of compounds that were tested. There were 40. And we'd see how well, of the hits that were identified this way, how do they rank according to the, to GLIDE SP, right? Ideally, they'd be right at the very top, right? So that would be a fantastic enrichment in that buy list. In reality, you can see they don't do very well. So GLIDE SP is represented in the, it's the bottom corner of that figure, and you could see, I mean, this is awful. This is much, much worse than random. They test GLIDE XP, which is different flavor of docking, do a little bit better. W-score, which is our water-informed docking method, is basically able to deliver basically random performance. And then finally, if you look at ABFEP performance, right, look at that fantastic early enrichment. What that means, what does that translate to? Well, we could have, instead of ordering 40 compounds to find those hits, we could have ordered 17. So a pretty dramatic decrease in the number of compounds we have to test in order to find all of those same hits. So really, it's going to be that improvement in accuracy that we hope to drive better virtual screening outcomes. So the cost, though, is unavoidable, right? So one of the reasons that docking is popular still is that it's cheap. And GLIDE isn't the cheapest solution out there, but compared to an FEP calculation, it's basically pennies. So for $10, right? So you got $10 in compute cost on the cloud to spend. You can dock 120,000 ligands with GLIDE. That's not too bad. Or you can get one data point from ABFEP, right? So very, very different. So you can imagine it'd be very hard to drive. I'm going to simply replace docking with FEP. Well, not really, right? I mean, not unless you intend to, spend, massive amounts of money just on compute alone. So you think next, well, this is what I'm going to do. I'm going to dock the compounds, and then I'm going to take the very best compounds. I'm just going to run that through ABFEP, right? That's a cascade. We're all familiar with these hierarchical methods. Save the good stuff for the very bottom. But look at this, right? Here's a plot of the docking score versus the FEP, right? I mean, if you're using this to pick, you're not going to do very well in terms of prioritizing which compounds will give you the best FEP score. So instead, right, the natural thing to do is think about, OK, well, what about machine learning? Right? How about basically reducing the cost here per each data point with active learning? And active learning, I'm grateful that Pat didn't talk about this topic because so it's left it for me. The concept is essentially you've got something that's very expensive to compute, right? Whatever that data point is, in our case it's FEP, and we're going to basically attempt, initially we're going to do sort of a random initialization of that model, select some points, run the expensive calculation, train the model, and then we're going to use a combination of, depending on balance of exploration and exploitation, we're going to try and improve that model so it gets better and better. And the idea is the cost of an ML evaluation is orders of magnitude smaller. And so we're able to, with the same budget, screen the entire library with some pseudo FEP level of accuracy. So next question, well, can you really learn it, this function? And if the answer is you can learn it, but it requires lots and lots and lots of value, a huge training set that we can't afford to build, that's not very helpful. So what you're looking at here is a very expensive test. I told you it was $10, right, for each one of these data points. So we did a test where we accumulated 6,000 of these predictions. And we asked, well, how well could a machine learning model sort of in one-shot training do with various sizes of training sets? And you can see what the enrichment looks like. So thankfully, we're seeing pretty significant enrichment. But it's not a small expense still, right? 500, right, 1,000. We see a critical jump. And interestingly enough, this is repeated actually across projects. It's about when you get to 2,000 compounds in your training set that you really start to see this really next level of performance. But it does drop off after about that point. And so here I'll just flip through some slides really quickly. So you can see, really critically, I'm not such a big believer in this R-squared metric. Really what we care about here, and this is how you're going to drive your project forward, is am I shifting that ABFEP free energy distribution, right? Am I actually identifying more potent compounds? And just flip through these. Point out that it does seem to converge at some point, although this is still quite expensive. So next, I'm going to show an example where we start with the active learning, and we get a model that looks like this, which is really, really crummy. Right? It's R squared of 0.1. Right? Can we actually improve that? Well, if we go to a second iteration, really the first iteration of real active learning, we see a real jump in predictive power. And you can see we really start to be able to learn that model. In fact, we're doing better with this iteration 1 model of explaining that the compounds and the initial random batch. And here's the shift I was talking about. So you start in the light blue, again, kind of crummy, centered around minus 7.5. But by the time we go through multiple rounds, we've really shifted. And if we believe here that this is going to correlate with experimental binding affinity, that's exactly the kind of shift we want to see. OK, so there's not much left to go through here. I will point out there's a lot of flexibility in how you define in terms of your acquisition function, how aggressively, how greedy we want to be. If we see, for example, that generally successive iterations with our greedy strategy reduces the diversity, the compound diversity, which is a concern, but that we can cover that. We publish this method in our active learning glide paper, balanced uncertainty metric, balanced diversity metric for selecting compounds in active learning, and you can see we can recover basically the initial diversity of that first set. And finally, I'll just point out, here's another example here, which sort of compares the two selection strategies. One, which is using machine learning, right, where we're sampling that training set from a very large number, 100,000 of the best docking compounds. And on the right, we're basically using our old strategy of let's use a cascade and sort of pick the compounds at the very, very top of the docking funnel. And you can see we've pretty dramatically shifted that outcome. And finally, here's a prospective application. This is from a recent fragment screen. We confirmed a 50% hit rate, testing only 42 fragment hits, balancing a whole bunch of other properties. And there was a lot of other machine learning behind this. But really the workhorse here is being able to deploy that absolute binding and overcoming what is essentially nothing, no real information from the docking scoring function. So with that, I'll stop. And here are some of the people at Schrodinger responsible Thank you for your time.
Thank you, Stephen. I think we have time for maybe 1 or 2 questions. So the other Stephen.
Hey, Steve Kearns from Relay. Just hearkening back to some of Pat's comments on automated chemical design, can you comment on how much post-filtering and visual inspection was done in your active learning iterations, or is this entirely machine-driven selection at this point?
Entirely machine-driven selection. I will say we're not yet at the point where, and I hope we can at some point, where I can say, here's the output of our algorithm. Let's go buy these compounds. At Schrodinger even, we still want, teams want to be able to inspect and have some input. But as far as, the algorithm itself, it's 100% free of human intervention.
Thanks.
Is there an online question?
Yeah, so it's related to your slide 7. Basically, the question is, how is Glide SP so much worse than chance? And also, does that mean that Schrodinger SP and XP are not reliable for virtual screening? And does that mean that one has to purchase FEP for the job?
Yeah, I like that question a lot. So the key to remember here is this is in a screen of 3.8 million compounds, right? So Glide SP already identified 13 hits, right? So if it had, if it was a random performance, we wouldn't have gotten those 13 hits, right? The real question here, and that was the motivation of this original paper, was, okay, how could you boost it further with rescoring? And which of these rescoring methods would be most effective? And what I can tell you from this one data point is that sticking to another docking method, only gets you so far. We think you really need, free energy calculations to do a lot better.
Great.
Hi, I'm Mrinal from Broad Institute. So you compared your absolute binding energy calculation with your docking methods, and absolute binding energy calculations are computationally very expensive. So have you tried some path-based, metadynamics-based calculations which are not as expensive as absolute binding energy and something intermediate between?
Yes, this is a very active research area for us right now. So as you can imagine, there's a lot of people at Schrodinger who would like to make this less expensive to run, and so there's a few different ways we've explored doing this. One of them is something called, you can think of it as sort of dynamic, lambda windows, essentially. So we're allowing the, if we do enough test calculations, can we sort of figure out, how long we need to run the simulation, how many lambda windows do we need, can we use a cheaper protocol and get similar results? In several projects, for example, we were able to reduce simulation time to from 5 nanoseconds to 1 with essentially very little loss in performance. I wouldn't expect that to be generalizable. I think you have to do tests. But I think protocol optimization is definitely possible. And yes, we are actually exploring something quite similar, which is looking at, is that initial binding mode stable, right? And we maybe don't need to throw all of this compute to figure that out. So yeah.
Great, thank you so much, Stephen.